Okay, so we should be live now. So, hi everyone. Welcome to this episode of Sussex Visions. Just really quick again, a reminder about how the format works. We're gonna now hear the talk by Christian. During the talk, you can post the questions on the YouTube chat feature. You need to be logged in for that, just in case. And then at the very end of the Zoom link after the questions part, I'm going to put the Zoom link that we currently have on there so you can actually join yourself and then talk to Christian in person. Okay, and now I'm excited to introduce our speaker for this week. Christian is originally from Chile, and then he did his PhD in Canada at the University of Alberta. He then went on to do his first postdoc in Munich, where we briefly overlapped actually very early during my undergrad days. And now he's back in Alberta. And already back in my time in Munich, I learned that he's very, very enthusiastic about understanding the bird visual system. So it's going to be really cool to hear what he's up to these days. Yeah, take it away.
Oh, yeah. Well, thank you very much, Michael, for the invitation and the introduction. And yeah, I'm excited to tell a little bit of what I have been doing, some of the areas of what I have been doing. Let me share here. And so today, uh, all right. Does that look good now? You can see the uh,
Yeah, should be fine.
you can see the presentation now.
Yes, that works.
All right. So today, I'll be talking about optic flow and the neural circuits in the avian brain that detect optic flow and how we think this might get transformed uh, into motor outputs to control avian flight, right? And this is part of a long-standing project now for more than 10 years that is a sort of collaboration between two labs, between the Wiley lab, and that's Doug Wiley, who is my supervisor. I'm a research associate in his lab in the University of Alberta in Edmonton. And and so this project is joined between the Wiley lab here in Alberta and the Altshuler lab. That's Doug Altshuler, who is a professor at the zoology department in UBC in Vancouver. And these two labs came together because the Wiley lab has been studying for many years how birds, the visual system of birds, the evolution of the visual system, and particularly how they detect and use optic flow. And then the Altshuler lab, I think, was very interesting in flight, and they have been studying the biomechanics of flight in birds. But eventually, they decided that it would be a good idea to join together and study how maybe birds use, like how their brain uh, to control flight, right? And particularly, an immediate target was optic flow, and I'll tell you what optic flow is. So, optic flow is the motion that happens across the entire retina when animals move, right? So this is sort of a cell generation generated motion stimulus that happens in all animals inevitably when they move across the world, right? And here Gibson in 54 already started speculating that this was very important to control locomotion in animals, right? And um, interestingly, Gibson used a bird to show this, right? So this is a drawing from Gibson from 1954, and here's a fly flying forward, and these arrows are showing essentially the pattern of optic flow that this bird is going to get across the retina, right? So essentially, the arrows are showing that in front, the bird is getting this sort of expanding optic flow, and on the side is getting this sort of right horizontal optic flow, and in the back some contraction, right? And so, and Gibson already speculated, like I said, that this is going to be important or essential for the control of locomotion, right? And this is in fact true. So people had shown that this is true in insects, for example. This experiment made by Srinivas in 1991. Uh, they essentially took some insects, some bees, I think, and put them in a tunnel, right? And in this tunnel, there's some stripes, right, to generate optic flow, right? So as this fly flies forward, it's going to get optic flow from both sides. And so if you do this experiment, you put the same stripes in both sides. The birds, the bees, the insects tend to fly right in the middle of the tunnel and right in the middle of the tunnel. And this is because they're trying to balance optic flow, right? They want to get the exact same amount of optic flow on both sides, right? But if you change the optic flow, so if you change the stripe patterns, now you put some horizontal stripes that are going to generate sort of no optic flow, then the bee flies much closer to this wall. And this is because it's trying to balance, right? It's trying to generate more optic flow by moving closer, right, in an attempt to balance optic flow, right? And similarly, if you move one of these walls, right, so now you put this stripe pattern and you make it move forward, right, at a speed that is similar to the B speed, essentially, making generate less optic flow for this bee also moves closer to the wall, right? So this shows that these insects are detecting optic flow and using it to control their locomotion, their flight, right? And uh, in 2015, people, Schniffer and also the same group, showed that in budgies, right? So in these parrots, the same is true, right? So you do pretty much the same experiments. You make them fly through a tunnel with some vertical stripes that are going to generate optic flow, and when you put them the same stripes, they're balanced. The budgie flies right through the middle. If you move the bars, right, to sort of diminish the amount of optic flows, the budgies tend to fly faster, right? So in this case, both sides have sort of less optic flow for this budgie, and it's trying to generate more optic flow by accelerating. And if you do the opposite, they fly slower, right? So they have some sort of expectation of optic flow related to how fast they're flying, and then they try to match this depending on what you're doing, right? And so again, it's showing that these birds are detecting optic flow and they're using it to control their locomotion. And of course, a very interesting thing is about birds is that there's 10,000 species of birds, and they come in many different ecologies, and they do different things. And that means that they fly in different ways, and they use fly in different ways for migration. For some birds, hunt on the fly, right? Like a falcon or like an insect-eating bird, like a swift. But a very interesting birds, and one that we are interested on that we have been studying, are hummingbirds, right? And hummingbirds are unique in that are the only group of birds that can generate sustained hovering, right? So some other birds can generate a little bit of hovering, but hummingbirds do what you see here, which is that they can hover in one space, right, in one spot for a sustained amount of time. Another very unique things hummingbirds do is what you can see here is that hummingbird flies briefly backwards, and that's a unique characteristic of hummingbirds. And so, and of course, hummingbirds have this sort of very unique adaptations in their physiology and anatomy to be able to do this, the shape of their wings, etc. And so this is interesting, and I'm going to talk more a little bit about what we have learned from hummingbirds. And one thing that Doug Altshuler showed early is that hummingbirds also use optic flow to control their hovering, right? And so here you're gonna see a video. Here is a control, sort of there's no manipulation of the optic flow. This is a nectar, right? A pipette filled with nectar. And this has been sort of quickly deep lab cup, right? So the hummingbird is labeled with a dot that you're gonna see just so you can see it better. And as you can see, here it comes, and as you can see, this hummingbird, just as it's staying very stable, right? So it's very good at staying very, very still while eating the nectar, right? It's coming in, and as you can see, it's staying perfectly still while doing this. But if you mess with their optic flow, so here you have this expanding spiral, right? So this expanding pattern of optic flow in front of the hummingbird. You can see that the hummingbird is drifting backwards, right? Um, and you can see here maybe better, but as you see, the hummingbird can't stay as still as this one is actually constantly drifting backwards. So again, it's telling you that this hummingbird, in their attempt to stay steady in one spot, is also using optic flow, right? Okay, so Where in the brain of birds is optic flow processed? And this is something that people have been studying since the 80s, and what Doug Wiley has been interested in for many years. And so here's the brain of a pigeon, and this is from a paper from the Chilean group that has studied the visual system of birds for a long time. And I love this picture because it just showed you how visual birds are. We say always that birds are visual animals. Well, this is the eye of a pigeon, and you can see just the size of it compared to the brain of this pigeon, right? And one structure that you can see very clearly on this brain, and this is a picture from sort of from behind, right? This is the spinal cord and the brainstem, but you can see this sort of large structure here, which is the optic tectum, which is homologous to the superior colliculus in mammals. And as you can see, it's very large. In fact, these structures receive direct inputs from the retina, and it receives about 90% of the inputs from the retina. But birds, like any other vertebrate, has multiple visual pathways that go from the retina to different parts of the brain, right? So they go to the optic tectum, but then they go to the thalamus, for example, the dorsal thalamus. Would be very much homologous to the classic DLG and visual cortex pathways in mammals, right? In birds, this is called the visual wools. You have the ventral thalamus, GLB. And then you have also the tectum, which has both, give rise to both ascending and descending pathways, including one to the thalamus and then the entopallium, which is so another part in the forebrain of birds. And so another sort of ascending cortical pathway. But in birds, there's two retinorecipient nuclei, right, in the pretectum called LM and MBR that are specialized in detecting optic flow, right? And we have known this for a while, since the 80s, when people started recording from this nuclei, okay? And so LM it stands for nucleus lentiformis mesencephalic, and this is in the pretectum right next to the optic tectum, and it receives direct inputs from the retina. And then you have the nucleus of the basal optic root, MBR, which is as known as the accessory optic system. And these nucleus are highly conserved in all vertebrates, at least in all amniotes at the very least, I would say. You find an MBR and an LM in all of them. In mammals, the homologue of this nucleus are the nucleus of the optic tract that is in the pretectum and homologous to LM, and then the medial and lateral and maybe dorsal terminal nuclei. So there's two or three, depends on which mammalian species, they're homologous to MBR. Okay, and people have known for a while that this nucleus, both in all amniotes, detect optic flow, and the cells in this nucleus respond to optic flow. And I'll show you some examples in a little bit. And so, but where does this pathway start, right? So they start, of course, in the retina. And so already in the retina, we know there are cells that are direction selective, and that they give sort of start these pathways to this nucleus, right? So in mammals, from a series of work of different people, we know that the medial terminal nucleus, the homologous to MBR, receives input from what are called the on direction selectivity ganglion cells. And here's just one example of one of these on direction selectivity ganglion cells in a mouse from Sun et al. And as you can see, this cell, right, if you show an object or a stimulus moving in different directions, right, here are plotted in this sort of polar plot. And as you can see, this cell responds very strongly in one direction, right? Here, sort of downward. And then it's actually sort of inhibit on the other direction, or responds very little on the opposite direction, okay? And this is what one of these cells sort of look like in a mammal. Now, in birds, no one has really recorded from direction selective cells, but we do know which cells project to this nuclei, to MBR and LM, okay? And so we can sort of assume that these cells must be, as in mammals, direction selective, okay? Particularly in MBR, the homologous to MTN, which receives input exclusively from on direction selectivity ganglion cells, if you put an injection of a tracer there, you find that the cells that project here are a special kind of ganglion cell called displaced ganglion cells, right? So, and these are from the original paper that discovered this in the 80s from Fitte et al. which was from Harvey Carton's lab. Um, and as you can see, these ganglion cells are very unique in that they are not in the ganglion cell layer, which is shown here in a pigeon, but they actually reside right in the amacrine cell layer, right? And so for a while, people thought that cells in MBR and that MBR receive inputs from displaced ganglion cells, but this was the only place that received inputs from displaced ganglion cells. Now, some time ago now, we did some tracing and we repeated these sort of injections in MBR, but we also injected a retrograde tracer in LM, the other optic flow nuclei in the pretectum. And we found, to the surprise of us, that when you inject a tracer, a retrograde tracer, in this case a red tracer in LM, you also get some displaced ganglion cells, right? And so, and of course, in MBR, this is very much true. Notice that when you inject in LM, you get both normal ganglion cells right in the ganglion cell layer and displaced ganglion cells. In the case of MBR, you almost exclusively get displaced ganglion cells. And this was the same case where we did injections of different colors, and we managed to get this beautiful picture where we have the displaced ganglion cells, right? This is the amacrine cell layer. My mouse is moving slowly, but as you can see, you even got this like side-by-side displaced ganglion cells that are in the amacrine cell layer, and that one projects to MBR and the other one projects to LM, right? So we think that the situation, and this is speculation, but we think that the situation here must be similar to mammals in that MBR receives input from displaced ganglion cells, which we could assume maybe that they're on this direction selectivity ganglion cells, and LM receives both from these ones and from on-off this direction selectivity ganglion cells, which in mammals have been shown to project to NOT. Okay. And actually, we then later, because we were doing injections in the LM of hummingbirds and on zebrafinches, we reproduced this result in these two other species. So you can see here in zebrafinches, right, we inject a retrograde tracer, which was BDA 3000, and which works beautifully anterograde, but also retrogradely. And so you get these very nice displaced ganglion cells, and the same is true also even in the hummingbird. So this seems to be a sort of universal, perhaps, to all birds, that displaced ganglion cells project both to MBR and to LM. Okay, so, but an interesting thing about hummingbirds particularly is that hovering requires a lot of stabilization, right? So hummingbirds are flying, staying steady on one side, and this is because they need to extract nectar from flowers, as you can see here. And hummingbirds, if you look at the head of this bird, it's very steady, right? It's staying in one place as it's extracting nectar from this flower, and this despite, you know, beating its wings at a very high rate and moving fast, but they're still. So one thing that we have thought over time and that we have to look at it is how is, oh, sorry about that, is how is the optic flow system, right, detection system of hummingbirds might be specialized to sort of match this requirement of sort of some extra stabilization or some very sharp stabilization. And one of the first evidence that there's something special perhaps about the optic flow system of hummingbirds came from the Wiley lab and from Andrew Iwanuk, who is now who was a postdoc at that time in 2007 in the Wiley lab, is now a professor in the University of Lethbridge, and he did a comparative study where he measured the size, the volume of LM, right, and MBR, these optic flow detecting nuclei in birds, and compare the size of this nuclei, the relative volume, to other birds, right? And here you just have this a coronal section to the LM of a hummingbird. This is LM, this is on a pigeon. But when you look at the volume, right? So this is the log of the brain volume, this is the log of LM volume, and in magenta you have hummingbirds. And as you can see, the volume of this nuclei is much larger relative to brain size than most other birds, right? Particularly here, these are swift actually. So you can see that for birds that are very similar size, um, hummingbirds have LMs that are much, much larger in volume, right? And so at that time, and interestingly, this is only true for LM, but not for MBR, right? The MBR of hummingbird is actually of a sort of similar size to all other birds. And so at that time, already started speculating that this has to be probably related to a sort of increased need to stabilize images and stabilize the whole body when hovering for hummingbirds. Now, recently, we did to further sort of to further try to understand if there's more difference in the visual system between hummingbirds and other birds that don't hover. We studied the retinal projection in this species, right? And so we did an injection of a tracer, a fluorescent tracer. In this case, is a colorotoxin, and we inject it in the eye. You expect a few days, and then you can look at the brain, and this labels all the terminals from the retina. These are coronal sections in a zebrafinch, and this is in Anna's hummingbird. And you can see that this is the optic tectum. And as you can see, the optic tectum has a topographic representation of the retina. And so here you can see that our injection filled the entire retina, right? Perhaps with a little spot missing there, but you have most of the retina. It's filled with the tracer, and so now we can compare the targets of the retina in this species. And I must say, we didn't find really large difference in terms of the targets. So the targets in hummingbird, the same regions that I showed you before, in the thalamus, in the dorsal, and the ventral thalamus, they're all very similar. So there's no very large difference. But we did find one interesting difference, and that was precisely an LM, right? And so when we look at LM in detail, here again, this is a coronal section to a zebrafinch LM. This is a hummingbird LM. One thing that is going to become important on this talk that you should know now is that LM is divided in two subdivisions. There's a medial subdivision that you can see here, and there's a lateral subdivision here. The same, you can see it here in the hummingbird. And what we found is that when you look at the terminals from the retina in this region, so you can see here terminals, this is the ventral thalamus, this is LM full of terminals, same here for the hummingbird. But when we look at the intensity, right, of the labeling, we found that there's a difference between zebrafinches and hummingbirds. So, and particularly in how intensely you get labeled the lateral subdivision of a lamb compared to the medial subdivision, right? So if you look at the zebrafinch here, this is the medial subdivision, this is the lateral. Here's some close-up, and if you measure and standardize the intensity of this terminal labeling, you get that LML is somehow more strongly labeled, but there's not a huge difference between these two nuclei. But when you look at the hummingbirds, and you can see here in the picture, it almost looked like there's no labeling on the other nucleus, but this is because it was very hard to get a picture that can show both, because the labeling in LML, right, on this lateral subdivision LM is very, very intense. This is standardized, and so this is several standard deviations from sort of the labeling on the other nucleus, much, much brighter than the other nuclei, right? So LMLs seems to receive this sort of particularly set of terminals that label much more intensely. And so we measure these, we sort of measure the intensity of labeling in all these different retinorecipient nucleus, right? LML, LMM, MBR, and also some pretectal and thalamic targets just to compare it. And what you see is that in the zebrafinch, the MBR has this sort of more intense labeling. But when you look at the Hannah's hummingbird, is the lateral LM that has a very, very intense labeling, right? And so this is, again, a little bit, it's speculative, but what we think is happening is that this intense labeling of the terminals and this sort of different terminals probably comes from the displaced ganglion cells. And we think what this is perhaps showing is that in hummingbirds, the LML is receiving a strong input from displaced ganglion cells. like NBR, and this is why this labeling is much more intense compared to several finches, where it's probably the element is receiving displaced ganglion cells too, but there is a sort of difference and perhaps a larger projection of displaced ganglion cells to LML in hummingbirds. And we think this is very interesting because, as I'm going to show you, this might explain, A, could explain the difference in size of LM, but it also might explain other difference we have found in LML. I mean, in the LM of hummingbirds, particularly in the response, as I'm going to show you next. And so one of the first things that this collaboration between the two labs did some years ago now was starting recording uh, the response to optic flow of cells in MBR and LM of hummingbirds and several finches and compared it to pigeons, right? And this was work that was done largely by Andrea Gaete, who was a postdoc at that time at the lab of Doug Altshuler and who's now a professor at the Royal Veterinary College in London. And so she very skillfully managed to put hummingbirds, in a recording setup and managed to record cells from LM in hummingbirds. And she found something very interesting. So here's an example of an LM cell responding to optic flow. So this is one cell in the LM of a hummingbird. And what is happening here is that the stimulus that is being presented, right, that is a big, large bars moving across the eye, allows the contralateral eye of this bird. And every some seconds, right, this stimulus is moving in different directions, right? And then stopping and then moving in different directions. And the arrow here is showing that direction. And you can see that these cells have a high sort of spontaneous rate during, right, when there's no stimulus. And as you can see, this cell A is inhibited in some direction. In this case, this cell is inhibited when the movement is downward and forward. But then this cell responds more and more and responds particularly on a sort of down backwards movement, right? And then you can see that in other directions either doesn't respond or responds just a little bit. And then when you go back to the sort of opposite direction, you get this sort of inhibition, right? So this is how cells in both LM and MBR behave. And so a very interesting thing that Andrea Gaete found was that when you look at the response of the population of cells in LM in pigeons and in several finches, you find that the cells on the LM of birds in general have as a population a preference for temporonasal motion, right? So this is motion that goes right from the back to the beak, right? And so these are, each dot is the preferred direction of a different cells, right? And so as a population, these cells tend to prefer one direction. And as you can see, the same is true for the zebrafinch. This has been shown, this was shown by Doug Wiley many years ago. This was Dre doing it in the zebrafinch. But then when you plot the hummingbirds, you see that there is no sort of population preference, right? You find cells, you find in the LM of hummingbird a sort of equal representation of preference for all directions, right? And so there's a difference already here, right? So here it's showing that there's a difference in the sort of response to optic flow between hummingbirds and other birds. And later, Graham Smith and also with Dre and other people also further analyze the response of LM neurons in hummingbirds and compare it to other animals. And what they did is, in this case, they actually compare different temporal response to different temporal frequencies, right? So how fast these bars are moving, and also to different spatial frequencies. So how together are these bars, right? So you can test this sort of different combinations of both temporal and spatial frequency for these cells. And here's an example of an LM cells. And as you can see, these cells are sort of tightly tuned to a sort of specific spatial frequency and a temporal frequency. And the ratio between these two is velocity, essentially, right? How fast the stimuli is moving. And so essentially, the cells are tightly tuned to one specific, to some velocity. And when you compare it, right, these are the sort of this is the maximum, right? The peak tuning of different cells in different species. So you have hummingbirds, finches, and pigeons in different colors. And this is right different cells, and the peak tuning it's mapped here. And as you can see, there's a difference between species, right? So hummingbirds prefer lower spatial frequencies and high temporal frequencies. And essentially, in that hummingbird cells prefer a much faster velocity than other species, right? And so there's a significant difference here where hummingbirds prefer faster moving optic flow than the other species. And this is an LM. Interestingly, when you do the same in MBR, you find that there's not really many difference. And there's no difference on the directional tunings. So this is sort of the population directional tunings that I showed you before. For the three species, this is in MBR. And MBR, as I was shown before for pigeons by Doug Wiley, the preference are sort of up, down, and backwards, right? And so you have this sort of three directions that are mostly represented, and this is true for all three species, and there's not a lot of difference. If you do the same as we did before with the LM, looking at the sort of preference for temporal and spatial frequency, you don't see many difference between the species, right? So the response on LM are very different. You have different representation of directional tuning, and also you have a sort of preference for faster moving elements. How am I doing on time? All right, I'm gonna move a little bit faster because now I want to tell you about the other part that I was trying to tell you, where we were trying to figure out how this optic flow might get transformed into motor outputs to control locomotion. And so an important characteristic of both of these two nucleus, LM and MBR, is that they project to the cerebellum. And this is almost unique, or not unique. This is different from mammals, I would say. It's true for reptiles that both LM and MBR project directly to the cerebellum. In mammals, the projection is not direct. It goes through the ponting area. But in birds, both LM and MBR project to the cerebellum, and they project to two areas of the cerebellum. So this is a coronal section through the pigeon cerebellum. Pigeons have foliated cerebellums like mammals, and they have different folias that are named in Roman numerals, right? And so this is anterior, this is posterior, and what you get is that in birds, the MBR, particularly and LML, the lateral subdivision, they project to the back of the cerebellum to folia 9, CD, and 10, and this is what is known as the vestibular cerebellum, right? They also receive this area also receive an indirect optic flow input through the inferior olive, right? So LM and MBR also project directly to the inferior olive, and the areas in the inferior olive that receive this input project to the vestibular cerebellum, to the same areas. Now, additionally, LMM, right? So the medial subdivision of LM and a little bit of MBR project to what is known as the oculomotor cerebellum, which are folias 8 to 6. And this is from a work already from Janelle Pacan, who was a grad student at Doug's lab some time ago. So one thing we know, and we know this from studies of early gene activation from Fenders in 2008, and this is from a hummingbird, is that the cerebellum is active during flight, and particularly you can show that these areas that receive optic flow, right? So the vestibular cerebellum here and folias and the oculomotor cerebellum are highly active during flight. So this is essentially they took a hummingbird, made it hover and fly for half an hour, and then they did some early gene activity, and you get this sort of nice activation of these two areas, right? And so we think we know that this area, the vestibular cerebellum, and this is because we know this in birds and also in mammals, that this area is related to oculomotor or collicomotor, so neck reflex to stabilize the image, right? So this is known in many animals. So we think that this kind of sort of fast stabilization of the neck and eyes is probably happening through this area. But we have become very interested on what this might be doing, right? So this is the other main target of optic flow to the cerebellum, but we don't know exactly what this is doing. But we think we can get an idea from the sort of inputs to this area, and I'm going to talk a little bit about that. And so in pigeons and from the work of Doug Wiley for many years, he has shown that the vestibulocerebellum responds very nicely to optic flow. You have two areas that respond to optic flow. The lateral area, the flocculus, cells respond strongly to rotation, and you have these sort of parallel zones of response to rotation in different axes. And then on the medial, you have this very nice response to translation, and you have in different stripes and different parasagittal stripes of the cerebellum, you have response to different types of optic flow, right? Contraction, expansion, ascent, and descent. And because these areas ultimately project to the vestibular nuclei and to eye and neck premotor and motor areas, is that we know that these areas are pretty much involved in stabilization of and the optokinetic response and coliomotor reflex and stabilizing the neck and head of birds. So then, right, what I show you here. But an interesting question to us was, what is this oculomotor cerebellum is doing, right? And we think what we can get an idea of what this area is doing from the different input that it's getting, right? Because this area is not only receiving optic flow from a lamp, as I show you, but is receiving both visual and other inputs from the forebrain and from other nucleus, other visual nucleus, right? And so one important target that we have been looking at is SPM in the pretectum and the pontine nuclei in the brainstem. And so birds have this nuclei SPM called the medial spiriform nucleus, and this receives inputs from the two main outputs of the forebrain, the wools and the arcopalium. And essentially, these are the outputs of the two main ascending visual pathways I showed you at the very beginning, right? The one that goes through the tectum and the one that goes through the thalamus. And both SPM and the pontine nuclei receive these inputs and they project to the oculomotor cerebellum. And additionally, of course, you have the input from a lamp. And so over the last few years, I have been studying the organization of these inputs to the oculomotor cerebellum to get an idea of how these inputs are organized and what this might tell us about the function of this area. And I'm running, I think, out of time, so I'm going to try to run a little bit faster about this. But essentially, one thing we did, for example, is that we injected tracers, retrograde tracers, in different zones of the cerebellum of birds. And one thing that you should know is that the cerebellum of birds and of all vertebrates is organized in parasagittal stripes, right? And so this means that both the inputs and the outputs of the cerebellum are different as you move from the medial to the lateral part of the cerebellum, right? This is a coronal section to the oculomotor of the oculomotor cerebellum of a pigeon, and you have these different zones that have different letter names, A1, A2, C, D, E, right? And so we put tracers in these different areas, and you get these nicely retrograde labeled cells in LM. Notice that for the oculomotor cerebellum, the projection to the cerebellum arises from the medial LM and not from the lateral LM. But we found something very interesting is that there's a difference in sort of the ipsilateral or bilateral inputs from a lamp for the different zones, right? So we found that this zone A1, which is in the middle of the cerebellum, receives equal inputs from the right and the left LM, but the lateral zones, the zones C and A2, are actually receiving inputs that are almost exclusively contralateral, right? So there's a sort of slightly different sort of type of optic flow you're getting in different areas of the cerebellum, right? In the middle, you're getting binocular optic flow. In the side, you're getting monocular optic flow. Additionally, like I said, we look at the inputs from SPM and the pontine nuclei, which are receiving inputs from both the tectofugal pathway, right? So this is the arcopalium. This is one of the outputs of the forebrain in the cerebellum, in the forebrain of birds. And this is sending projections to SPM and the pontine nucleus from the tectofugal pathway, which detects local motion or looming. but also from touch from the head. Additionally, the visual wools, right, projects to the pontine nucleus and SPM, and also the somatomotor wools, which is the equivalent of the somatosensory cortex in mammals. And this area is also sending descending projections. And interestingly, in birds, the somatosensory representation of the body is sort of separate from the head. The head is represented here in the arcopalium and nucleus basalis, but the whole body is represented on these anterior wools, okay? And so we have been doing injections of anterograde traces on these different areas, both in the visual and rostral wools, but also in the different sensory areas of the arcopalium. And to make a long story short, we found that both SPM and the pontine nuclei have this sort of segregation of inputs so that the medial part of SPM receive input from the wools and the lateral part from the arcopalium. And within this, the sort of visual and somatosensory areas are also separated between medial and lateral. And the same is true for the pontine nuclei, where the medial pontine nuclei receives input from the somatomotor wools, and the lateral pontine receives input from the visual arcopalium, right? So from the tectofugal pathway. So there's a separation of inputs, as you can see here in this sort of nice picture, where the inputs from the rostral wools are shown here, the visual right are right next to it, but this all go to the medial part of SPM and not to the lateral part, which receives from the arcopalium. And we found that this is also true for other inputs. So we have traced with Andrea Gaeta to inputs to the medial pontine nuclei from LM and GLV, the ventral thalamus, but also from the optic tectum. This large visual area received most of the inputs from the retina. And again, to make a long story short, we found that these inputs are also segregated between the lateral and the pontine and medial pontine nuclei, so that the medial pontine nuclei receives input from LM and GLV, and the lateral pontine receives inputs from the tectum, right? So again, the sort of different visual inputs and different somatosensory inputs are segregated between these pontine nuclei that, as I showed you before, in different pontine nucleus. And the very interesting thing we found is that when we look in the same experiments I showed you before in LM, so when we inject traces on the different zones of the cerebellum, we found that the inputs from this medial and lateral SPM and from the medial and lateral pontine are also segregated in different zones, right? And importantly, we found that the projection from the, right, and this is so that the lateral pontine nuclei and the lateral SPM project to the medial area of the cerebellum, and the medial SPM and the medial pontine project to the medial sides to the lateral parts of the cerebellum, right? And importantly, we also find, like in LM, a difference between bilateral and contralateral inputs, so that the lateral SPM and the lateral PL, as is shown here, they essentially project bilaterally to the medial zone, but the medial SPM and the medial pontine project almost exclusively contralaterally. And so when you put all these things together, you essentially get these sort of parallel pathways that have optic flow, but also other visual and somatosensory inputs to the cerebellum. And they form a sort of parallel pathways, right? The medial SPM and the medial pontine receive inputs from some areas, right? Like the visual wools. And then the lateral pontine and the lateral SPM receive inputs from other visual areas and other, both in the forebrain and in other areas. And so the inputs are segregated and the inputs are segregated and the outputs are segregated so that they finish in different zones of the cerebellum, right? And so essentially you get these parallel sensory motor pathways to zone A1, the medial part of the cerebellum. You get a bilateral optic flow and bilateral tectofugal pathway. And on the other side, you get contralateral optic flow and contralateral visual and somatosensory inputs. And I'm going to finish quickly just to tell you that what we found very interesting is that this has been shown a long time ago that these different areas of the cerebellum project to different cerebellar nuclei and different vestibular nuclei. And these are the, right, so these are the outputs that Purkinje cells in these different zones, which are the outputs of the cerebellum, projects to different nucleus depending on which area they are. And so this creates not only two different parallel pathways to the cerebellum, but also in the inputs, but also on the outputs, right? And so so we think that these sort of parallel inputs to the cerebellum are then translating to two parallel outputs, one to the medial cerebellar nuclei and one to the lateral cerebellum nuclei. And these nuclei have these sort of very distinct projections. The medial cerebellar nuclei has sort of descending projections to the reticular formation to all, and here's where you have a lot of cells that control locomotion and also to the spinal cord. So there's this sort of strong descending projection, while the lateral cerebellar nuclei project forward, particularly to nucleus ruber. And so we think that these have different functions, right? Where optic flow, bilateral optic flow on one side is being used to control forward flight. And this is mediated by this medial cerebellar nuclei that projects directly to these sort of motor areas that control locomotion, right? And so we have bilateral optic flow. We have a bilateral optic flow from the vestibular cerebellum. We have bilateral optic flow here, and this projects heavily to locomotor areas. On the other hand, we think that CVL and this lateral cerebellum might be involved in maneuvering, and this is because this lateral cerebellum receives only input from one side of the right monocular optic flow and other monocular visual and somatosensory inputs. And importantly, this lateral cerebellum nuclei projects to the nucleus ruber, which in all vertebrates sends long descending projections to the spinal cord to control both the hands and the tail and the legs. And so we think that this might be a way where birds are directly controlling the hands, the wings to maneuver and using optic flow for maneuvering. Also, this is all part of a classic sort of sensory motor loop that exists in most vertebrates. So in all, we have two circuits for flight control, right, that use optic flow. One to the medial cerebellar nuclei that uses binocular optic flow and probably to control steady flow and things like landing. And on the other hand, we have one that is probably controlling turning and maneuvering and through projections to the nucleus ruber and to other areas. And so that's about it. Future directions, there's a lot of exciting things happening in the both labs. We're doing recordings while flying, manipulating circuits with optogenetics, and we're also studying the organization of the pathways from the cerebellar nucleus to the brainstem and the spinal cord. And that's it. Thank you to everyone, of course, and Andrea Gaete and everyone who's been involved in all of these studies, the two Dougs, et cetera, and everyone else. Thank you very much. And thank you for the invitation.
Thanks for the talk. There was a really cool project on mapping all of these gestalization circuits, really exciting. There's also a lot of questions, so I'm just going to go through them chronologically. The first couple are all by Tom. The first one is noticing how he noticed how in birds the presumed direction selective cells don't stratify in like one or two neat monolayers like it would be in mammals. Any insights on what that means functionally or what starboard amacrine cells look like in birds, or if that's known?
Sorry, so oh, you mean because like the ones the
They did,
actual ganglion cells, they're
yeah,
not. Oh,
they didn't like stratify neatly in like
that's
one
a
layer or
good
two
point, and
layers.
I haven't thought about that. That's a good point. Uh, yeah, I don't know exactly what I would tell you. Like I said, yeah, we were talking about this before, but yeah, because no one has recorded from these cells, so yeah, maybe it means that there's more different types. It's possible the injections in a lamb can leak into the optic tract. So
Mm-hmm.
it's also possible that in some cases that we're not always right labeling only the ones to project to a lamp. Although we had, so maybe we need to look at the specific cases. I think we had some cases that were better than others in that the injection was very, very restricted and very in the middle of a lamp. But
Mm-hmm.
you have to go through the optic tract that goes to the tectum to get to a lamp. So it's hard to completely discard the possibility that those could be some of them just project to the tectum or other areas.
Okay, yeah, makes sense. I was about to ask that anyways, just for clarification. Okay, so then the second question is like kind of two questions is that baseline activity seemed to be really huge in those guys. Is that a typical feature of visual neurons in hummingbirds? Because apparently we see something similar in chickens as well.
You mean the spontaneous activity?
Yeah, yeah, the spontaneous, the
That's
base.
very true for a lamb, but I don't think it's so true for other ones. I think in the tectum, I don't think the tectum is, yeah, that's how we find them essentially. Because
Mhm,
when you go down, it's like you sometimes you can see other visual stuff, right? As you're
mhm.
going down, depending on the angle, that's how you immediately know you're in a lamb because suddenly there's so much noise and there's so much spontaneous activity. So I would say that that's a very lamb and MBOR thing, but not
Okay.
necessarily on other visual areas.
But then that could also be a retina thing, would make sense, right? Because there's lots of input coming from there. So high. Okay. Interesting.
Yeah.
Um, and then this has been also quite discussed in the chat between Tom and Henrike, but he asked, is it like there is the idea out there that birds use less energy per spike than other species? What's your opinion on that?
Oh, yeah, I guess that was the work of Honor Gunter-Kuhn, and I forget her name. But yeah, they show that in the brain in general, you get this sort of much efficient use of energy.
Hmm.
Um, I don't know what my take on that is. I think I believe them. I don't have reason to doubt that result. I think it's interesting, and yeah, it's interesting to think how, and it might explain other things like why birds have so many neurons, right? Right? They have a much higher density
Hmm.
than mammals, and so it might explain things like that. But yeah, it's an interesting result and one that I don't know if that might be true for the retina too. Maybe that's why the retina is not so vascularized and because it doesn't need, that's why they have
Hmm.
such a weird like pecting and the sort of lack of actual direct vascularization. I don't know. Yeah, but it's an interesting result.
The other one is also that's actually something that's been bothering me since my PhD as well. What's with the vertical direction selectivity bias in brains across species? Seems counterintuitive for flight control.
Vertical. No, but it's not vertical. It's
It's the
but you mean
temporal
in
to nasal, right? But like just in
yeah,
general,
because it's
there's this
backwards. Yeah,
bias.
but it is, and I always yeah, and that's only in yeah, and I guess I didn't mention that, but that bias that exists in pigeons and finches is true
Hmm.
for all vertebrates. Yeah, so you go
Yeah.
to a mammal NOT and they also have the same yeah, sort of bias. But all the other directions are represented in MBR. So it's
Hmm.
not that the brain as a whole is biased. It's just that the directions are separated. That's how I have seen it always. That, all the three other directions are sort of represented in MBR, and this one is represented in LM or the terminal nuclei. Why that one in particular? For a while we speculated, I think Andy speculated that it was because hummingbirds fly backwards, right? And as
Hmm.
you can see with that experiment with the optic flow, right? Like they have to sort of balance on that directions, right? Like if they're sleeping backwards, right? They have to fly forward. So in hummingbirds might make sense why yeah, why the LM is larger, that's how we make it to make sense. But then why there's no bias in the LM of hummingbirds makes, I know, we don't know, we're not sure why these things are true.
Yeah, we had the similar thingy in my PhD lab. We did it in frogs, and they also have this bias in
Oh,
LM.
okay, so
Actually, if you only do one eye, then actually there's a bias in the optokinetic reflex as well. But I also don't know.
okay, yeah, cool.
Okay.
So you recorded from a lamb in frogs.
Yeah.
Cool.
Okay. So then the next one is a bit open-ended by Pranav Seth. Um, if we could get robust AAV expression in specific RGC types in birds, which part of the optic flow pathway would you be most excited to map?
Oh, if you could get, yeah.
Yeah, if you could like do
Well,
imaging with, I guess.
definitely get, if we could map the displaced ganglion cells, then that would definitely answer my question, are the
Hmm.
displaced ganglion cells projecting only to LML and not, right?
Yeah.
I would be very excited to get that answer. Like, are these displaced ganglion cells to LM going to one nuclei or both, right?
Yeah.
Because then that would, right, that would,
Oh, like if the same, yeah.
yeah, because then because that starts a whole different pathway, right? Because LML projects to the vestibular cerebellum, right? So then it's telling you that they're doing something different, perhaps, than the other sort of on-off, right? So maybe the on-off are going to LMM, and those are going to the oculomotor cerebellum. So then we're getting this sort of very distinct pathway. So then we could actually confirm that maybe, right? Because the other hypothesis would be that that's not true, and that the displaced ganglion cells project to both, and right, and so then maybe you have a mix of things going happening.
Yeah. Could also
But
be that
definitely,
it comes from like
yeah,
non-direction selective. Like I think in mouse, that's a thing that like you generate direction selectivity from other inputs. But yeah.
yeah.
Okay. Just a quick note for anyone who actually wants to talk to Christian in the Zoom. I'm going to post a link now. I'm going to leave the link in there. You can join, and I'll let everybody in once the stream ends. There are a few more questions though. Um, One is from Anna Vladec. It's a big jump from 3 to 4 directions of optic flow motion to complex retraction, expansion, etc. in the cerebellum. Do you have any idea how this optic flow information from LM and the NBOR are integrated with each other in the cerebellum?
Yeah, I think there's some idea that I think it's definitely you can make those cells, but by combining all this sort of cells, right? But it's true that yeah, it's an interesting transformation where you think about it because you have this direction selectivity, large cells, and then now you have this sort of translation, rotation. But yeah, I feel like I haven't thought about this in a while, but I think it I think if you read some of Doug's paper, this is definitely something we have thought about in the past.
Mhm.
That how that transformation happened. But definitely, yeah, it's a good question because
Okay, so
definitely, yeah,
there's some models out there, and
I think so. I think there's definitely
yeah.
some because the same is sort of true in mammals. There's an impressive convergence there where if you go to the flocculus and the nautilus of mammals, you find the sort of same zones and the same sort of responses. So yeah, there's definitely some models out there of how you get there.
Okay, and then I think that's the last question, also from Pranav Seth again. Could avian complex spectral tuning ability influence motion pathways, especially given that they might fly earlier or late?
Oh,
So like
that's a
with
great
spectral,
question.
yeah.
That's a great question. And I don't think anyone has sort of gone there. That sort of interaction between sort of spectrum, color, and
Mhm.
and optic flow, for example, right? Like I think in the very sort of classic way people used to look at this, some people for a long time they were trying to figure out where is the color vision, like what responds to color. And there's some old paper by the Chilean guys and by Maturana where they go like,
Mhm.
oh yeah, GLV, this ventral tectum uh, nuclear responds to colors or something. But it's my impression that more and more that, and especially from looking at Tom talk sometimes, that color, is not how we think about color and the
Yeah.
fact that, like, oh, we're distinguishing red and whatever in objects, but it's probably, yeah, I don't know exactly how color vision is going to work, but it will be very interesting to start combining these things because I think they are always being studied separately, right? People go to
Hmm.
the optic
Yeah.
tectum and go, oh yeah, the cells are very sensitive to motion, and you have all this characterization about motion, and the same with optic flow. But it's always black and white. It's always about contrast, and no, I don't think anyone has sort of, yeah, what happens if you start varying the colors and yeah,
That
and
also gets
spectrums.
at some point really hard to like design your stimuli, record all of this in like one session, but yeah, cool to figure that out, I guess. Smooth
But
along.
definitely the optic flow, in that sense, the optic flow is a good one because they're
Mhm.
very robust,
Yeah,
right? And we know
exactly.
exactly
Yeah.
what they sort of have always looked like. So it will be a good place to maybe do that and sort of see what happens. I'll ask Doug. I wonder if he has an idea about this, of what color might be doing. Yeah.
Okay. Cool. That was it for questions. Again, for all the viewers, I put the link into the YouTube chat if you want to join in and maybe chat with us. I'm going to leave it up for a while. And then in a couple of minutes, I'm going to end the stream, which will also take down the link. So join up if you want to chat. Yeah. No, that was really cool. Like all the mappings and all the mapping, the injections. Seems hard to do as well because you need to like do you have like a good stare tactic atlas for
Yeah, we have for the pigeon, for both the finch and I must say that the people who do those injections, so Dre,
Mhm.
and there was, oh my God, I'm gonna blank on her name, but there was a student in Doug's lab who was extremely skillful at injecting all the different nucleus. Uh, and it was because there's no stare attacks. So I think a lot of that was happening was finding a lamb, which we, like, so that was established. Like we now know, you put the bird in the finch in certain angle and you know where a lamb is. And from that, I think they started moving around and finding GLV, and the tectum is easy to inject because it's big.
Yeah.
My injections haven't been that bad because a lot of those are big targets, like the wolves and the arcopalium, and yeah. So, but yeah, I do love. I think I really like how I think the anatomy can really sort of direct where you're going and what and sort of because now we have this sort of hypothesis, and they're all based on sort of mapping these pathways, right? And there's now a student in Doug's lab in Doug Alshuler lab that is starting to record from the oculomotor cerebellum and then from the cerebellar nuclei, right? Because we want to see how you might get different responses, right, down
Yep.
this pathway.
Okay, I think I'm gonna end the stream then. So far, yeah. And then we can chat a bit more before we end things.
Sure. No,
Okay, thanks everybody for joining, dropping by to listen to the talk. Thanks a lot
th-
to
no,
you
thank you.
for
Thank you for the
presenting your work.
invitation. It was really
Yeah,
good.
cool. Okay, we are now offline again. One thing that I'm also kind of interested in